Let's talk about carbohydrates today. The topic is chemical nature and functions of main organic compounds of organisms. So under that, carbohydrates. Most abundant group of organic compounds on Earth is carbohydrates. So the most abundant group of organic compounds is going to be carbohydrates. What is water then? Water is going to be the most abundant inorganic molecule, right? Here we're talking about organic, containing carbon, right? Clear? So we have to talk about that. So most abundant group of organic compound on Earth is going to be carbohydrates. Major elemental composition is carbon, hydrogen, and oxygen. So why did they say major elemental composition is going to be carbon, hydrogen, and oxygen? Why did they say major? Because sometimes there can be phosphorus and nitrogen. Why? Because there are derivatives of carbohydrates. For example, if you take phosphoglyceraldehyde is going to be a derivative of triose. In phosphoglyceraldehyde, we have phosphorus. And if you take N-acetylglucosamine, we have nitrogen. So these are going to be derivatives of monosaccharides. Hope it's clear. So the major elemental composition is going to be carbon, hydrogen, and oxygen. Hydrates of carbon contain the same proportion of hydrogen is to oxygen, which is equal to 2:1 as in water. Now you know in water, we have H2O, this is the structure of water. So we have 2:1 to be the hydrogen is to oxygen ratio, right? And if you look at the general formula of carbohydrates, if you look at the general formula of carbohydrates, it's going to be CxH2OY. So can you see here, I have H2 times Y and O times Y. That means the ratio between hydrogen to oxygen is going to be 2:1 because it's in brackets and it's being multiplied by Y. Hope it's clear. So the general formula is going to be CxH2OY. Very important, CxH2OY. Three major groups of carbohydrates are monosaccharides, disaccharides, and polysaccharides. So there are three major groups of carbohydrates. Now what are the three major groups? Basic one, monosaccharides. Second basic, right, disaccharides. And then the large ones, they're going to be polysaccharides. Generally, carbohydrates include sugars. Now what do you mean when you talk about sugars? You talk about monosaccharides and disaccharides. You talk about sugars, you talk about monosaccharides and disaccharides. And polysaccharides are not considered to be sugars, right? So we talk about the reasons. So polysaccharides, they are a different category. They're not considered as sugars, but we consider monosaccharides and disaccharides as sugars. Hope it's clear. Monosaccharides. The simplest form of carbohydrates. The word simplest. So you have the simplest form of carbohydrates having the general molecular formula as CH2ON. So we have CH2ON to be the general molecular formula. Very important. Once again, you can see the ratio of hydrogen to oxygen to be 2:1. Now, it's the simplest form of carbohydrates because monosaccharides, they get together and form polysaccharides and disaccharides. For example, glucose gets together with fructose, which is another monosaccharide, to form sucrose. And also, if you consider glucose molecules, right, they can get together, they can polymerize and form cellulose, which is going to be a polymer. Now you can understand that, right? So they're the simplest form of carbohydrates having the general molecular formula as CH2ON. Where carbon varies from 3 to 7. So the carbon number here varies from 3 to 7, right? All monosaccharides are reducing sugars. Very critical point. All monosaccharides are reducing sugars. Now, why do you say that they are all reducing sugars? Now, listen, I'll explain this like this. If we have a group like this, it's called an aldehyde group. So suppose I have carbon here, hydrogen, hydrogen, and the rest of the glucose molecule, right? Just giving a rough representation. So if I have a carbonyl carbon, now this is called the carbonyl carbon attached to the oxygen like this. carbonyl carbon, and I have hydrogen. This is called an aldehyde group. So this group is called an aldehyde group. Aldehyde group. Actually, this group can oxidize itself like so. I'm not representing the rest of the molecule. It can oxidize itself like this. It can become the carboxylate ion, eventually carboxylic acid. So you can see that instead of hydrogen, I'm getting oxygen, which means I'm oxidizing carbon, right? So actually, if you talk about oxidation numbers, now because of carbon here and carbon here, there's no distribution of oxidation numbers. Because carbon is attached to oxygen, which is an electronegative element, carbon gets +2 due to oxygen. And due to hydrogen, now hydrogen has a lower electronegativity than carbon. Therefore, carbon gets -1 and hydrogen gets +1. Therefore, the oxidation number of carbon overall is going to be +3, +1 here, because +2 from oxygen and -1 due to hydrogen. Therefore, +2 minus 1 gives you +1. What about this carbon here? This carbon is going to have an oxidation number of +3, right? Because 3 bonds with oxygen. Therefore, there's going to be an increase in oxidation number, which means this glucose molecule, for example, containing the aldehyde group, is going to oxidize itself, which means it's going to reduce somebody else, because we talk about redox reactions. When you talk about redox reactions contain oxidation reduction reactions, which means oxidation reaction is followed by the reduction reaction. They happen together. We can't have one reaction which is oxidation reaction just happening alone. They happen together. Hope it's clear. Right, so all monosaccharides are reducing sugars. All, word all is really important. All monosaccharides are reducing sugars. They're water soluble and they occur in crystalline form. All monosaccharides are reducing sugars, point number 1. Point number 2, they are water soluble. And point number 3, they occur in crystalline form. So you can see them as crystals, and you can see that they're water soluble. Glucose, for example, is water soluble, by experience. So according to the number of carbon atoms, oops, I just went through down. According to the number of carbon atoms, they're named as 3 carbon. Now, if it's 3 carbon, we say triose. Triose. So triose is the 3 carbon group. What are the examples of triose? Glyceraldehydes. Now, what about glyceraldehydes? We have phosphoglyceraldehyde, which is a derivative of triose, right? Now, glyceraldehydes, actually these are found in the process of photosynthesis and respiration. Glyceraldehyde is a 3 carbon group, right? It's a 3 carbon compound. But phosphoglyceraldehyde, it's getting a phosphate group in addition to the carbons and hydrogens and oxygens, right? So then what happens is phosphoglyceraldehyde is going to be a derivative of triose because we are getting phosphorus. For carbon, we have tetroses. For carbon group is going to be tetrose, right? So the example, that is erythrose. Erythrose is found rarely in nature. Actually, erythrose is found in blood too, but the content is less, right? So the erythrose is going to be rare in nature, right? Hope it's understood. Right, we have 5 carbon sugars. Pentose. So pentoses, if we talk about pentoses, we have ribose, deoxyribose, ribulose. Right, so ribose is found in RNA. Now when you talk about RNA is made up of ribonucleotides. In ribonucleotides, if you consider ribonucleotides, we have the sugar group, the nitrogenous base, and the phosphate group. The sugar group, which is represented by ribose. Deoxyribose is the same story, but here the sugar group is represented by deoxyribose. So ribulose is found in what? The processes of photosynthesis, you can see. Right, let's talk about these things later. But we talk about RUBP, which is the derivative of ribulose. Now RUBP is a derivative of ribulose. Why? Because it has phosphorus. So it's going to be a derivative. When you talk about 6 carbon sugars, right, monosaccharides, we can talk about hexoses. And the examples are going to be glucose, fructose, and galactose. Now, according to the carbonyl group, they're classified as aldoses and ketoses. Now, what do you mean by the carbonyl group? we have carbon and oxygen attached like this. This is a carbonyl group. So if I have R here, and then if I have hydrogen here, this is going to be the aldehyde group. This is the carbonyl group, right? Carbonyl group, and then this is going to be the aldehyde group, aldehyde, right? Now I have the carbonyl carbon, but it's not attached to hydrogens. It's not attached to hydrogens. What can you say? It's not attached to hydrogens. This is going to be the ketone group, ketone, right? So in aldehydes, there's an attachment to hydrogen. So let's see, according to the type of carbonyl group, they're classified as aldoses and ketoses. So aldoses are they glucose and galactose. Ketoses, the example is going to be fructose, very important. They're testing these in MCQs. So they may ask which of the following is a ketose sugar. So you have to give fructose as the correct answer. Right, let's proceed on to the next one. Yeah, all right, there's a point like this. Can you see there's a point like all monosaccharides are reducing sugars? Now, why do you accept that? All monosaccharides are reducing sugars. Now, actually, to be able to act as a reducing sugar, there should be a qualification. Now, what's the qualification? You should be able to do this. To act as a reducing sugar, we should have the aldehyde group to be able to reduce others. That means to oxidize itself. We discussed about this. But what about this ketone sugar, fructose? It actually doesn't have this aldehyde group. Then what can you say? Now, how is it going to act as a reducing agent? This is because now fructose doesn't actually contain an aldehyde group. But now what happens is fructose can be isomerized to distinct aldoses. It can isomerize itself to distinct aldoses. That's the reason. Therefore, then again, you can regain the reducing capability. It can reduce others and oxidize itself. Right. So we discussed this. So you can see the solid form of glucose and the aqueous form of glucose. Actually, this should be the aldehyde group. I have to have the hydrogen here. This is going to be the aldehyde group. So you can see the aldehyde group. So therefore, this is going to be glucose. And look at the ketone sugar. Keto sugar. If you look at that, the solid form of fructose can be seen, and you can see the keto group here. And one important thing to note is that the aqueous form of fructose is represented to be cyclic. But if you talk about the solid form, it's going to be acyclic. It's not a cycle. Not a cyclic form. If you look at glucose, the solid form is going to be acyclic, but the aqueous form is going to be cyclic, ring-like. You can see in aqueous media, some monosaccharides are in ring form. But the word some is very important. In aqueous media, some monosaccharides are in ring form. Not all, right? Okay, so we come to disaccharides now. This is a different color. Disaccharides. So they are sugars formed by joining two monosaccharides by glycosidic bond. How are they formed? They're formed by joining two monosaccharides through what? A glycosidic bond. Right, now let's see. How are they formed? Now look at this. On the right side, you can see the formation of maltose. On the left side, you can see the formation of sucrose. Let's see the formation of maltose because they have represented how it's really formed. We can see glucose molecule 1 and glucose molecule 2. Now, if you consider glucose molecule 1, the OH from it and the hydrogen from the other glucose molecule, they leave as water. So OH from one glucose and the hydrogen from the other glucose, or OH from one monosaccharide and the hydrogen from the other monosaccharide, they leave as water. And then the bond formed is known as the glycosidic bond, which you can see here. This is the glycosidic bond. So the glycosidic bond is formed by replacing the OH and the hydrogen that left as water. Hope it's clear. So the glycosidic bond is formed by removal of a water molecule. So removal of a water molecule, we saw that. from two adjacent monosaccharides. How? By condensation reaction, right? So glycosidic bond is formed by the removal of a water molecule. We saw that. We saw that a water molecule was removed from two adjacent monosaccharides by a condensation reaction. Water molecule is formed from OH group of one monosaccharide and the hydrogen from the joining monosaccharide molecule. We saw that too, right? Very clearly we saw the OH from one glucose and the hydrogen from the other glucose. It's a very important one. Glucose and glucose, they combine and undergoes a condensation reaction to form maltose. So we have glucose and glucose forming maltose. If you have glucose and fructose, they combine to form sucrose. If you have glucose and galactose, they combine to form lactose. So glucose, glucose, maltose, glucose, fructose, very important. Sucrose. Glucose and galactose, lactose, right? Remember this. This is going to be a very critical point now. Maltose and lactose are reducing sugars. So if you consider maltose and if you consider lactose, both maltose and lactose, they're reducing sugars, which means they can reduce others and oxidize themselves. But sucrose is going to be a non-reducing sugar. Really important point. They are testing this in MCQs and structured essays. So sucrose is a non-reducing sugar. It can't reduce others. So the conception, what's the conception that disaccharides are all reducing? It's wrong, because out of disaccharides, sucrose is a non-reducing sugar. Monosaccharides we discussed, and then we discussed disaccharides. Now we have to talk about polysaccharides. Polysaccharides, they are macromolecules and biopolymers. They're macromolecules and biopolymers. Macromolecules because they have a lot of atoms, therefore they are heavy. And biopolymers because biological polymers, these polymers are found in the biological environment, in biological living organisms. So what is a polymer? Monomers get together and form a polymer, right? So monomers get together and form a polymer. So here, for example, glucose monomers, you can represent these glucose molecules by these circles. Suppose glucose molecules are represented by the circles, and suppose this connection is represented in the bond, glycosidic bond. So glucose monomers are joined together by glycosidic bonds, and for example, they form cellulose, which is a polymer, right? So polymers are formed by monomerized. Polymers are formed by what? Combining monomers, right? So polysaccharides are made up of a few hundred to a few thousand monosaccharide subunits. Yeah, polysaccharides are made up of a few hundred to a few thousand monosaccharide subunits. So we have hundreds of thousands of monosaccharide subunits helping to form polysaccharides. Very important point coming up. Let's use a different pen. They're non-crystalline. So you can't see them as crystals. They're non-crystalline. And they're water insoluble. They do not dissolve in water. And they're not considered as sugars. Right? They're not considered as sugars. We saw that at the very beginning too. And they're not crystalline, therefore you can't consider them as sugars anyway. Some polysaccharides act as storage components. So some polysaccharides act as storage components, whereas others contribute to the structure of living organisms. So some polysaccharides are then acting as structural components. Based on their function, they're categorized as storage polysaccharides and they're classified as structural polysaccharides. Storage and structural, right? So let's see the examples of storage polysaccharides. Starch, starch is found in plants. We have store food. Glycogen is found in fungi and animals, right? To store food. So for example, now glucose, they combine together to form glycogen. Glycogen is actually stored in the liver and muscles. Now, for example, if you have a lower amount of sugar, if the sugar content in your blood is less, glycogen can be converted back into sugar, right? To make sugar more available. Structural, if we talk about structural sugars, polysaccharides, the examples are going to be cellulose, hemicellulose, and pectin. So the examples are going to be cellulose, hemicellulose, and pectin. Right. For example, cellulose makes up the structure of plant cell walls. Also, hemicellulose and pectin, they are also helping to contribute to form the structure of plant cell walls. Now, based on the architecture, they're categorized as linear forms and branched forms. So what are the linear forms? Cellulose and amylose. So cellulose and amylose, they're going to appear like this. They're linear. What are the branched forms? Glycogen, amylopectin, hemicellulose. So branched in the sense they appear like this. Branched. Right. Branched forms. So you can remember it like this. Amylopectin. Branch your pronunciation. Right? So you can branch it and pronounce. Amylopectin. Hemicellulose. Branch it and pronounce. Hemicellulose. Amylopectin. But then glycogen cannot be branched and pronounced, I accept, but you can remember at least amylopectin, hemicellulose, right? So glycogen is branched, remember that. Right, so we have major polysaccharides, their monomers and functions. So polysaccharide, for example, have starch. Starch is stored in plants, and the monomer that makes starch is glucose. Monomer of glycogen is going to be glucose as well. So monomer of glycogen is going to be glucose as well. And where is glycogen stored? It's stored in animals as well as in fungi. That's the point sometimes that you miss. It's found in fungi as well. Cellulose is made from glucose. It's found to be a component of the plant cell wall. Inulin, actually inulin is made up by polymerization of fructose monomers. So it's found in dahlia plants, in actually dahlia tubers more specifically. You can see, Google it up and see, right? So stored in tubers of dahlia. So you can say dahlia tubers. Pectin, where is pectin found? Pectin is found to be a component of the middle lamella of the plant cell wall. So it's found to be a component of the middle lamella of the plant cell wall, right? Now, middle lamella actually connects to primary cell walls of two different cells together. So galacturonic acid is going to be the monomer that contributes to form the pectin polysaccharide. So hemicellulose, if you consider hemicellulose, it's made up of pentose monomers, and it's found to be the component of plant cell walls, to be a component of the plant cell walls. Chitin, now very important, chitin is going to be a nitrogen containing polysaccharide. So we have what? Nitrogen based polysaccharide here. It contains nitrogen. And the monomer is going to be glucosamine. So you see the word amine, when you talk about the amine group, you talk about nitrogen in the amine group. So where is it found? It's found in fungal cell walls. Again, it's found in fungi. Where? This time it's going to be in fungal cell walls. And also in the exoskeleton of arthropods. Where in arthropods? In the exoskeleton of arthropods. It's actually helping it to be very solid and compact. Right, now let's see. Functions of carbohydrates. What are the functions? We talk about monosaccharides, energy source, glucose, for example. When you're running, you need glucose, right? Energy, because glucose is used up by cells. For example, we take glucose into our cells, we allow it for cellular respiration, aerobic cellular respiration, wherein we produce ATP, energy, right? It's energy source. Clear point. Building blocks of disaccharides and polysaccharides, very obvious. Monosaccharides are the ones that build disaccharides and polysaccharides. We saw that glucose and malts, glucose plus glucose produced maltose. Glucose molecules together, they polymerize to form cellulose, for example. Right. Disaccharides such as maltose, sucrose, and polysaccharides such as starch and glycogen, we went through all this. They're components of nucleotides. I said that. They're components of DNA and RNA. We discussed this. Disaccharides. When you consider disaccharides, storage sugar in milk. So lactose is going to be the storage sugar in milk. So in milk we can find the lactose storage sugar. Translocation in phloem. So if you have phloem, you transport food in phloem. So how is it transported? As sucrose, right? So sucrose is used in the translocation process in phloem. Storage sugar in sugarcane. Also, the storage sugar in sugarcane is going to be sucrose. Sucrose is also used as the storage sugar in sugarcane. Polysaccharides. There are storage polysaccharides as well as structural polysaccharides. Let's talk about storage polysaccharides. Starch stores glucose as an energy source where? In plants as well as in chlorophytes. So when they say plants only, it's a wrong statement because we have chlorophytes. Chlorophytes also. What are chlorophytes? Chlorophytes actually green algae. So in green algae we can find starch. Cellulose is also found in green algae. We'll talk about that later. Glycogen stores glucose as an energy source in animals and fungi. We went through this. We said glycogen is found right in animals and fungi. Inulin stores fructose as an energy source in daily tubers. We saw this too. Structural polysaccharides. Right, let's see. Structural polysaccharides. Cellulose in the cell walls of plants and chlorophytes. So we can see cellulose and starch in both plants and chlorophytes. So in both green algae and plants. Pectin is found in the middle lamella of plant tissues as magnesium and calcium pectins. We talk about this later under the topic of cell walls. So pectin is found in the middle lamella of plant tissues. Hemicellulose is also found in cell walls of plants. So hemicellulose is also found in cell walls of plants. We talked about this. Peptidoglycan, another polysaccharide, which is a structured polysaccharide, is found in the cell walls of prokaryotes. Is found in the cell walls of prokaryotes. Under prokaryotes, we talk about bacteria. So in bacteria, we can find peptidoglycan. Not all prokaryotes, like archaebacteria, they have different. They don't have peptidoglycan. So chitin in the cell walls of fungi, you can find chitin in the cell walls of fungi and in the exoskeleton of arthropods. In the exoskeleton of arthropods and in the cell walls of fungi, you can find chitin. We talked about this too. Right. Let's go through some questions now. Actually, I'm going to give you true or false statements, which will help you to understand this pattern of MCQs. Now, in MCQs, the basic idea is to see whether the statement is true or false. So instead of giving you long MCQ statements and questions, I can give you the basis of MCQs, which is true or false. So you have to tell me whether it's true or false. Let's try it out now. Right, carbohydrates. Let's see some questions. True or false? Statement 1. Pectin is made up of pentose sugar monomers and is found in the middle lamella of plant cell walls. Yeah, pectin is found in the middle lamella of plant cell walls. That is true, but it's not made up of pentose sugar monomers. It's made up of galacturonic acid monomers. Who is made up of pentose sugar monomers? The one who's made up of pentose sugar monomers, that table that you saw, was hemicellulose. So these are some tricky points. You confuse hemicellulose with pectin, and that's why this professors ask that frequently. Second one, both glycogen and chitin is found in fungi. True. Both glycogen and chitin is found in fungi. We saw that. Third one, fructose is not reduced sugar as it doesn't contain the aldehyde group. No, we understood, though it doesn't have the aldehyde group, it can convert itself to glucose, and therefore it's going to be a reducing sugar. So fructose is not a reducing sugar is wrong. It is a reducing sugar because we understood the statement that all monosaccharides are reducing sugars. Fourth one, all disaccharides are reducing sugars. Wrong, because we know that sucrose is a non-reducing sugar, lactose and maltose, they are reducing sugars. Fifth one, glycogen and cellulose are branched. Now, glycogen is branched, alright, but cellulose is going to be linear. Right, I hope you understood this topic. Let's meet up with a new video.